In a first for Africa, researchers are screening cancer tissue samples to find existing drugs that could be repurposed as effective chemotherapies for people of African descent.
When I first heard about drug repurposing and also actually setting up a tumor growth facility where one can really personalize chemotherapeutic drug choices, I was actually really excited because the drug repurposing is a fantastic way of re-looking at existing drugs and also affordable drugs to make them work for us in a new way. We then certainly thought that this will be a great partnership because, of course, the basic scientists need tumor to actually work from. And of course, we have no laboratory facilities or laboratory experience at all. So almost all patients with pelvic cancer, uterine and ovarian cancer, will then first actually have surgery. So they'll have an operation which is either diagnostic and we take biopsies, etc., or if possible, we will remove the tumor.
So if a patient consents to participate, then they undergo the surgery and their biopsy is donated to the study. When the surgery has been completed, they store the sample in a fridge that is in the gynae oncology unit to then go through to Steve Biko and collect the specimen and bring it to CSIR. So for each patient, we're able to screen 88 drugs and identify through a viability assay which drug is more effective for that particular patient's tumor and the cells that we isolated from that patient biopsy. The number of clinical trials being performed in cancer in Africa is almost zero. There is big paucity of data from the African continent. So we are coming from a drug repurposing approach where from the drugs that are in the market, are we able to identify drugs that are more effective in our population? So you cut down the drug discovery process from a 7-year process to a 3 to 4 year process because these drugs have already gone through the efficacy and toxicity studies. They are approved drugs, but they're not used in cancer.
The ultimate goal is to actually be able to analyze the tumor fresh while the patient is also still waiting for histology and recovering from surgery. We have about 4 to 6 weeks before we need and can actually start with chemotherapy. So during that time period, we would ideally want to actually find the correct drug mixture for that specific tumor. That will be our ultimate goal. So once we actually reach that, we could even use non-chemotherapeutic drugs and drugs which are usually at the moment used for another purpose, like an antibiotic, etc., and combine that with an existing chemotherapeutic drug because we will then know that for this specific tumor, that is going to be the effective combination.
So we're coming in and trying to bridge that gap, Can we perform the research that is needed for these clinicians to be able to make better decisions to aid the patient outcomes? So we want to give these patients options.